All right, next up, we want to discuss the molecules of life. So for this section, you're going to want to be able to list the 4 classes of organic molecules found in cells and then describe the processes by which those organic molecules are assembled and disassembled. So, it was once thought that all of the molecules of cells must contain some sort of vital force that differentiated them from the molecules of non-living things. So because of this, chemistry was broken into the divisions of organic, chemistry of living things, and inorganic, the chemistry of non-living things. We no longer recognize this idea of a vital force to differentiate between the 2 molecule types. However, we have kept this division for our own understanding. We now identify organic molecules as those which contain both hydrogen and carbon atoms. There are only 4 classes of organic molecules that exist in living organisms: carbohydrates, lipids, proteins, and nucleic acids. But despite there being only 4 classes of biomolecules, the diversity of life is possible because of the diversity of molecules molecules which exist within these categories. So in order to build or synthesize a macromolecule, we must use a dehydration reaction. It's called a dehydration reaction because it involves the removal of a hydrogen and a hydroxide group in order to join the 2 subunits together. When you remove an H and an OH, those can combine together to form HOH. In other words, 2 H's and an O, or H2O. Thus, the synthesis of biomolecules leads to the formation of water molecules. A dehydration reaction is just simply a chemical reaction in which those subunits are joined together in the formation of a covalent bond, and water is produced during the reaction. This is going to be used to connect monomers together to make polymers. So, for example, the formation of starch. Starch is a polymer, poly meaning many, from glucose. Glucose subunits are monomers, mono meaning one. You can kind of think of all of these different biomolecules as trains. They're great, big, long, complex molecules, but they are made by simply hooking these smaller subunits all together. Or if we wanted to break apart a train, we would unhook or break down that train one car at a time, one link at a time. So to build it up, we're going to string these subunits together through a dehydration reaction. To break them up, we're going to use a hydrolysis reaction. This is a chemical reaction in which a water molecule is added to break down a covalent bond. This is going to be used to break down polymers into monomers. So just as an example, the digestion of starch into glucose monomers. So here is the breakdown and the synthesis of macromolecules. What we have here is a great big long macromolecule that's made of a bunch of little subunits strung together. If we want to glue this unit to this unit, we're going to remove an OH from one side and a hydrogen from the other. Those 2 are going to go off and form water. Then the remaining subunits are going to be covalently bonded to each other. And the reason that we're calling this subunits and units is because this is the same process, whether we are talking about carbohydrate synthesis or protein or lipid synthesis. We're going to be using the same process of stringing these subunits together through dehydration reactions. Conversely, we're going to be breaking them apart with a hydrolysis reaction. And once again, it's going to look the same regardless of the biomolecule. In this case, we're going to add a water molecule back to, and we're going to basically kind of split that bond apart. We're going to add an oxygen group or a hydroxide group on one side and a hydrogen group on the other. Now, a lot of times students have a hard time remembering the difference between a dehydration reaction and a hydrolysis reaction. I kind of like to think of it as glue. If you remember from kids, elementary school, that Elmer's glue is not very sticky initially. When it first comes out of the bottle, it's wet, it's not very sticky. So in order to make things stick, you have to wait for that glue to dry, thus the dehydration. You got to dehydrate that glue to bond these 2 pieces of paper together. If you wanted to dissolve that glue, all you would need to do is add water to it. Hydrolysis, using water to lysis or to split apart. So I like to think of these bonds as Elmer's glue. You dehydrate it to stick these 2 together. You rehydrate it to break them apart. All right, that's it for section 2.3. You should just be able to explain the difference between organic and inorganic molecule, list those 4 classes of organic molecules, provide a very brief function for each of those, and then distinguish between dehydration reactions and hydrolysis reactions and what their uses are.